All right, good morning everyone, and thank you for joining me on this Q&A session. I'm going to go ahead and get started, and as people come in, feel free to interrupt me at any time and ask questions. I have some material prepared for you. Today, the title of my talk is Exploiting the Cell, the Life Cycles of Human Viruses. And before I get into that, I wanted to give a little more, oh hey guys, welcome to my Q&A session. I wanted to give some more background to myself so that we have some credibility for this discussion. So my name is Katelyn. I am a third-year PhD student at Princeton University in New Jersey, USA, which means in American standards, I'm about halfway done with my PhD, so wish me luck on the rest of it. I actually am originally from Colorado and graduated from the University of Colorado about 3 years ago, where I also studied biology, but I also studied history when I was in college. All right, so to get this discussion started, I really want to give an overview of what makes me tick as a scientist. So first of all, viruses are everywhere, like literally everywhere. So every day you come into contact with, I think it's like 10 million particles per minute. It's what it's thought to be. So like they're in the air everywhere, they're on surfaces that you touch, they're in the water that you drink, they're living in the people that you know. About 20% of the human genome is actually thought to be DNA from ancient viruses. So even yourself is filled with virus DNA. If you take a mill of ocean water, so like picture filling a thimble of ocean water. Hello, welcome to my Q&A. If you fill a thimble with water from the ocean, you will find 100 million virus particles in that thimble of water, which is just crazy. So also viruses, yeah, literally crazy. Viruses infect all types of organisms also. So of course they infect humans. We all know about like the flu and the common cold and dengue, stuff like that. But they're also infecting things like amoeba, single-celled eukaryotes. In fact, the biggest virus particles we know of are all infecting amoeba. They infect bacteria, so bacteriophage are actually the most abundant viruses on the planet. They infect cows, dogs, amphibians, herpes viruses, which are the viruses that I primarily study, which cause STDs. You guys all know of cold sores that you get on your lips. Hello. They actually evolved in dinosaurs about 300 million years ago, and today there are about 100 different species of herpes viruses that we know of, and only 8 of those infect humans. So the point of that I wanted to get about, the topic today is exploiting the cell, the life cycles of human viruses. And right now I'm just giving an overview of kind of my research interests and what motivates me, and hopefully that will prompt you guys to ask me some questions that are relevant to that. Feel free to ask anything, really. Even I'm a grad student, so anything about academia or grad school or vaccines or anything, really, I would be willing to answer. So basically, there are viruses everywhere, but you're not sick all the time, right? That's the weird part. Viruses only infect certain organisms and certain cell types, so their replication cycles are actually very specific. So even though you're constantly coming into contact with viruses, if you go swimming in the ocean, you're basically swimming in a virus soup. You don't come out dying of virus infection. And that's because viruses can only enter certain cell types and can only replicate within certain cell types. So one of the best examples of this is your skin is actually almost completely impermeable to virus infection. Viruses can touch your skin, but they can't actually enter it. The only way they could is if you had a cut, and then they could get in through your cut. Some viruses you have to inhale, so they have to be floating in the air for you to actually get sick, which is why we all don't want people to sneeze on other people, right? But even then, sometimes you inhale viruses, and the specific virus you inhaled can't actually infect your lung tissue, and they actually have to get into your bloodstream or something like that. So viruses, while they're everywhere, they do have very specific replication patterns, and the battle between the virus and the host is really an interesting subject to study. Because once they do finally enter your cells, they're master manipulators of your cells. So they take a cell that is programmed, for example, in your heart or like in your blood, something like that, that has a very specific job, and they completely reprogram it to become a virus factory. So your cell goes from being a normal functioning cell to now only producing viruses. which isn't ideal for your body, of course. Also, because of this, we have actually learned a ton about our biology just from studying viruses. So one example of this is cancer. The study of cancer and the study of virology is very intimately linked. Viruses such as polyomavirus can actually induce cancer phenotypes in cells. They have oncogenic genes. And by studying those viruses, we learn a lot about how cancer works as well. Another example is CRISPR technology, which is all the rage right now. I'm sure you guys saw the Chinese researcher a couple weeks ago that made CRISPR babies. It's a very controversial topic. But CRISPR is actually a bacterial immune system to combat viruses. And so that was originally discovered by people who were interested in bacteriophage and then found CRISPR. And then from there, that was realized that it could be engineered to do other things such as gene editing. So while there's all these things that you can study, the thing that I really like to think about is that your cells themselves are very highly organized. So that's both like organized throughout your body. So the cells that are in your brain, excuse me, are very different from the cells you would find in your intestines or in your heart or in your feet, for example, in your skeleton. But even beyond that spatial organization throughout your body, cells are also highly organized at the subcellular level. So this is referring to things like organelles, right? So your cells are not just a bag of protein and DNA floating around. They're very highly organized with membrane-bound compartments known as organelles, such as the mitochondria, the nucleus, the endoplasmic reticulum, things like that. And different cell types actually look very differently at the subcellular level. So while they all have certain organelles, they don't look the same. For example, in cells that are secreting a lot of protein, such as your liver cells or even your neurons that are secreting neurotransmitters a lot, have really active secretory systems. So the ER, the Golgi, endosomes, things like that, would look very different than a cell that's in stasis, like maybe a skeletal cell or something like that. So entire fields are devoted. I could discuss that. I was going to discuss that a little bit later with like the aspect of changing cell organization, but we should definitely talk about antibiotics. I'll write that down so we can get to it. Thank you for suggesting it. Okay. Okay, so entire fields are devoted to understanding how cells are organized. You have fields of developmental biology that want to understand how your cells are organized as an organism actually develops from a fetus to a full organism. You also have things like just cell biology that is still trying to figure out what are these organelles in the cell and what do they do and how do they change in different disease states during stress, things like that. You have things like cancer biology that are studying how when your cell becomes cancerous, what about its organization changes also. So the thing that really gets me, though, is that viruses already know all this. Like, they're not alive and they don't think, but they know so much more about our biology than we do. They enter your cell and they can completely reprogram it from this detailed organization to now becoming a virus factory. And that's what I really like to think about, because if we ourselves are struggling so much to understand how these processes work, we could just study a virus that already knows all this, qualify that, they don't think. But then by studying them, we can learn new things about how our cells work. And that is really what motivates me as a scientist. So my first question I like to think about is just, like we just talked about, how are cells organized at the subcellular level? Yeah, they are pretty smart, for an anthropomorphic term, of course. And then the second main thing I like to think about is how viruses manipulate this organization. And what can that teach us about our own biology? All right. Before I move on, are there any specific questions so far about what I've said? I'm going to go with a no on that. If you have any, please get them to me later. Yeah, well, that's okay. I'll just keep talking. I like talking. So, okay. So beyond all that, viruses rely on host machinery to replicate, right? So they can't just get into your cell and just replicate on their own if the host isn't helping out. They have to be able to reprogram the host to do this. So if we discover how this works, we can discover drugs that block this, and we can also gain insight into how our cells work normally. So this can go to your question, Lewis. So antibiotics, right? That's actually a term for combating bacterial infections, which some of them are pathogenic, and they work in very similar ways as viruses. But viruses, the drugs that generally stop their replication, are actually inhibiting ways that the virus interacts with their host. So one of the most common ways to combat virus infection is by blocking entry into the cell, because if the virus can't enter the cell at all, it obviously can't replicate because it can't get access to the host machinery that it needs to make new virus particles. So whenever a virus enters a host cell, viruses are coded in proteins, and this is both for membrane viruses and non-membrane viruses. But these proteins on the outside of the virus have to bind to a specific receptor on the cell. So this kind of goes back to what I was talking about previously, where viruses have very specific cell types that they can infect. So if, for example, herpes viruses or human cytomegalovirus, which is the virus that I study, it uses multiple receptors on cells to enter, but these receptors are only found in epithelial cells or fibroblast cells. So it can't just infect anywhere, definitely can't infect neurons. And so you can design drugs once you find out how that mechanism works of the receptor binding the viral protein. You can design drugs that would block that. So maybe you design a small molecule that fits right in the pocket where the virus binds, and it makes it unable to actually enter the cell to initiate the actual entry process. You use ribavirin on respiratory syncytial virus infants often? That's really interesting. We should talk about that at some point. So a lot of my studies focus more on once the virus enters the cell and what it does after that. But historically in virology, a lot of focus has been put on blocking entry and then also blocking exit, because if you can't get the virus out of the cell, it can't spread and perpetuate the infection. So another way that antibiotics can work, or in this case antiviral drugs or therapeutics, is by, once it gets in the cell, just stopping it from being able to replicate its genome. So the four main things that any virus has to do to establish a successful infection is 1, enter the cell, 2, replicate its genome, 3, assemble new virus particles, and 4, exit the cell or egress the cell so that it can go infect other cells. So if you can inhibit any one of those 4 pathways, you can stop the virus replication cycle and offer a successful therapeutic to an infected patient. So, for example, in herpes viruses, there's a kinase, which is a protein that transfers phosphorylation between other proteins that the virus relies on to keep its replication cycle going. And the only antiviral therapeutic that is known to work against herpes viruses, as of now, is a drug that inhibits that kinase. So herpes viruses actually are not treatable. You can give them therapeutics to put down the infection, but for example, any of you guys who have cold sores, you know they come back all the time, right? Which is gross. But you can't, so it depends on the virus. So in the case of a herpes virus, they have human cytomegalovirus, or HCV, actually has almost 200 genes. So it's not really possible to design one molecule that would inhibit all 200 of those gene products. You got to kind of pick the ones that are kind of hubs for the later parts of replication, which is a big challenge. So going back to what I was saying, for herpes viruses, there actually is no way to, no vaccines for herpes viruses. We don't know how to create attenuated strains that could give people resistance against them. So almost 100% of the world population is infected with the herpes virus. So people with cold sores, they're not rare sights, right? We see them all the time. So the interesting thing about that, right, is HSV1 is what causes cold sores, herpes simplex virus 1, and it stays latent in your neuronal cells that are connected to where you get the cold sore. So you might notice that you usually get cold sores in similar places, and that's because as it reactivates, it comes right back up at the same exact place and forms a cold sore in the same area. And so for that, right, you can get a topical treatment that's supposed to kind of limit the production of the cold sore, but you're going to get a cold sore again later. So there's no actual, good treatment for herpes viruses that would eliminate that overall. So great question. The kinase is specific to human cytomegalovirus, which is what I was talking about earlier. There's a drug called ganciclovir that can only inhibit the Hcv kinase. There are analogous drugs for HSV1 infection that also inhibit a viral kinase, but the kinase is a different protein, so the molecule works slightly differently. But that happened to be a good way to combat herpes virus infections. Talking about herpes is always fun. I go to the grocery store and I'm checking out, and people are like, "Oh, you go to Princeton, what do you do?" And I tell them that I work on herpes viruses, and they're like, like, gross, don't want to talk about that. Excellent question. Please ask more. Also an excellent question. So yes, so different types of herpes viruses have very different replication cycles. So if you missed my introduction earlier, this is what I love to talk about, so thank you for asking this. So human cytomegalovirus actually infects you originally by infecting your mucosal membranes, but it establishes latency, which means that it stays in your body forever, and it actually lives forever in your bone marrow. Whereas HSV1, like we were just talking about, infects you also originally through mucosal membranes, so it's a sexually transmitted disease often, but it stays latent in your neuronal cells that are connected to your skin. Then you have herpes viruses like KSHV, or Kaposi's sarcoma-associated herpes virus, which a lot of people forget is a herpes virus, but it infects mostly your skin, which is how it causes cancer. And then you have herpes viruses like Epstein-Barr virus, which causes mono, which is the disease that makes you really tired for months at a time. And that also eventually stays latent, but it actually stays latent in your white blood cells. So herpes viruses they replicate everywhere, but they're all grouped by their structural and genomics similarity. So they are part of the same family, even though they have such diverse replication cycles. Epstein-Barr virus, any herpes virus, you keep your whole life, but they go latent, which means they're dormant. So even though they're still within your DNA and your cells, they're not actually actively replicating. So a lot of your symptoms you'd get while they're actively replicating, you don't have. So in the case of mono, you originally get the horrible sleep deprivation issues, and you're exhausted and sick all the time. This usually goes on for a year. But then after that year, you still have the virus, but it's no longer active, so you don't have those symptoms. So they actually remain latent in the white blood cells, so they're not killing them. They just hang out in them. That's actually similar to how HIV, for example, hijacks your white blood cells and lives in them forever. I know I talk fast. I'm sorry. I'll try to slow down a bit. Okay. So great discussion about antiviral therapeutics there. If you guys have any more questions later, please continue them. So getting back to what... Yes, that is also an excellent question. I'm loving these questions. Okay. Yes, so this is actually one of the most confounding questions right now in all of virology, really, is how are viruses reactivated when they've been latent? And in the case of herpes viruses, it is known to be linked with stress. But I should say stress in quotes because, what is stress exactly, right? We all talk about how, you look really upset, have been having a hard time at work, you must be stressed. So there's that type of stress. There's also stress where, if you get infected with the common cold, your immune system is in stress and your body in general is in stress. Or maybe you've been eating horribly for a couple of weeks, you've been eating way too much cake or something like that. It is the holidays, so we can probably all relate with that. And your metabolism is stressed because it's not getting the nutrients that it needs. Oxidative stress can happen with different types of virus infections. Actually, herpes viruses HCMV specifically causes oxidative stress when it replicates. That's just a messed up, your mitochondria is not properly processing nutrients and creating energy. All that to say, so yes, stress is linked to reactivation of viruses. And the way that it's thought that happens is when a virus is latent, it's living in your cell kind of peacefully, and it's really happy being there as long as it can keep being perpetuated to the next generation, right? So the whole purpose of a virus is to keep replicating. So if it's just staying dormant in a cell, then it can keep moving along, But if it all of a sudden senses that your cell is starting to get stressed out, it gets worried that the organism that it's being hosted by is maybe in trouble and it might die. And if the cell that it's in dies, then the virus doesn't replicate, right? So it will reactivate then. And whenever you get a cold sore, for example, in HSV1, it's actually secreting virus through the cold sore. So it's hoping to get to a new host. And so that is how stress is thought to be linked to virus reactivation. Immunodepression is also definitely a way that the virus would feel stressed out. Though that's really more that the way your body can ensure that the virus infection doesn't go active again and remains latent is by constantly battling it with the immune system. So, of course, when your virus is latent in your cells or dormant, it occasionally goes through bursts of replicating again. But because your immune system has already seen this virus, it can recognize it really fast and keep it down so that it doesn't fully form a cold sore, to keep going back to that example. But if you're immunodepressed or immunocompromised, then it becomes a huge problem. So this is also, in the virus that I study the most, human cytomegalovirus, this is a huge problem, and actually specifically in pregnant women and also immunocompromised patients like HIV patients. So in some areas of the world, almost 90% of the human population is infected with HCV. And it's overall, for healthy people, it doesn't really do much. Like, you don't notice that you have it just lives in your cells. It doesn't even cause any recognizable symptoms. Maybe when you first get infected, there's some evidence that you get a fever and like symptoms of the common cold. But if you are a pregnant woman and you get a flare-up of HCV, it is actually the leading cause of birth defects in pregnant women that are not related to alcohol or drugs, which is crazy. I do have a recommendation for a book about viruses. I could send that to you after. It's actually written by a couple of professors in this department here at Princeton. I'm going to write down your name so I can send it to you. Okay. Thanks for asking. HCV is not related to Zika. They're very different viruses. Though, like we were talking about earlier, if you study both of those, you might find common pathways at the cell level that could lead towards the birth defect phenotype. And that's something that people are actively studying, of course. Where were we? We were talking about HCV. Yeah, okay. And then also in immunocompromised patients, such as HIV patients, when your immune system goes down, then HCV can flare up and start destroying all the cells that it's been replicating in. Their immune system has been keeping it under control. So actually, this is a general theme in many cases. So anybody who is immunocompromised, whether you got like a kidney transplant and so you needed to shut off your immune system so your body would accept it, or if you have a chronic viral infection like HIV, you usually don't die from like the HIV infection or from the kidney transplant. You actually die from all these silent viruses that you've had in you for a long time that sometimes we don't even know that you have because you never had symptoms. But when your immune system is shut off, then the virus can go haywire and can kill all of your cells, which is the main concern. I don't know exactly what you mean by mini explosions. You might have to describe that. Okay, so as you retype that question, so a lot of different viruses are known to actually require, besides entry, they require reorganizing your cell as they replicate. So that goes back to what I was talking about earlier, where cells are really highly organized, right? So if you mess up the localization of a mitochondria or of a peroxisome or the shape of the endoplasmic reticulum or the ER, you reprogram how those organelles are working and then overall how the cell is responding to the environment. So this happens all the time in development. For example, your organelles are constantly changing shape. They're changing the abundance of organelles to respond to different things like oxidative stress, like we talked about earlier. So viruses actually have a very common strategy where when they enter your cells, they don't just peacefully replicate throughout your normal organelle pathway. They really want to reprogram the cell to be the best virus factory that it can be. So they will often reorganize the entire subcellular landscape. So I'm talking about like lipid composition and organization, protein organization, small molecule organization such as calcium and reactive oxygen species and other small molecules. Metabolites will go different places, and your cell will start to look completely different throughout infection time. In succession. We should probably talk about that later because I'm still a little unclear on what exactly you're asking. So maybe you're talking about like when in the immune system, is that right? When cells are combating other infected cells? Keep trying to type here and clarify it, and I'll get to your question eventually. Okay, so over infection time, the organization of your cells might completely change. And actually, one of the best examples of this is the main virus I study, Hcv. And this cell is known as cytomegalovirus because cyto means cell and megalo means big. And when it replicates, it completely changes the size and the dimensions of the cell. You can see this in infected patient samples where you get a little hub of infected HCMV cells, and they're just huge compared to the normal cells. So if we can understand how viruses do this, so they've already entered, your cell is already in jeopardy, but they require this reorganization of the subcellular landscape, maybe we can understand how we can stop a virus infection after the virus has entered. So people for a long time have studied how to block virus entry, and for some viruses, blocking it works really well. It was pretty easy in quotations, but they did find small molecules that could block that. But it's really hard to stop the infection once it's already in the cell. So this is where my research comes in, and what I really want to know is what viruses require subcellular reorganization and how they do it, and if there are maybe conserved themes in how different viruses reorganize the cell. For example, influenza, dengue virus, a lot of herpes viruses, and other human pathogens actually break, for lack of a better term, break mitochondria during the replication cycles. So maybe there's a conserved way that viruses do this, that if we could discover it, we could engineer some type of small molecule inhibitor or protease inhibitor or something like that to stop the viruses from doing that. And then we would have a way to combat not only herpes viruses, but dengue and influenza and other human pathogens because of this conserved mechanism. Asking why viruses do something, everyone does. We would love to know why. But there seems to be no purpose to any of this, right? They evolved in intimate interactions with the host, and the ways that the virus can manipulate the host are just intimately linked to how the host actually works. And so it's not really so much of a why as a how they do it, because viruses are just trying to get in, replicate, and get out. Okay, so for example, going back to HCMV, it completely reorganizes the secretory organelles, so the endoplasmic reticulum, the Golgi, endosomes, other vesicles, into what's known as the viral assembly complex. And it also remodels the nucleus, so if you picture your favorite textbook picture of a cell, right, you have a circular nucleus, and you have the ER kind of next to it, and the Golgi somewhere, and vesicles coming in, which I'm a microscopist, and the cell looks nothing like that, but we're just going to stick with that picture for now. But the nucleus is circular, but during HCMV infection, it remodels it so it looks more like a kidney bean, and then right next to this kidney bean is this huge assembly complex. And this is just a giant viral factory that can whip out hundreds of virus particles in a couple of hours. The cell looks nothing like that normally, right? So as the virus is infected, it required this complete remodeling of organelles into that assembly complex. And if you perturb that from happening, you actually do stop virus infection. So there are multiple examples of if you inhibit the subcellular reorganization and the exploitation of the cell, the virus cannot replicate, it cannot spread, the infection ends. So really, if we can understand how the viruses are causing this, what are the cellular mechanisms that allow this to happen, we not only can find new ways to combat virus infection, but we will also have new understanding of how your cell is organized normally, or how that's changed in neurodegeneration or cancer, things like that, because at the core of all of those diseases is the cell and how the cell is organized and how it functions normally, and then in the disease it is perturbed and functions differently. So by studying any of those things, we would gain insight into all the other things. And that's part of the reason why I love studying this so much. Thank you. I think it's so interesting too. Also, I should note, thinking of infections in this like spatial temporal organization, so your spatial organization of your organelles and the temporal organization over time. So as a virus enters and replicates what's happening to the spatial organization, is a pretty new way of thinking about virus infections. So for a long time, virologists focused on, first of all, discovering viruses, because viruses weren't even really known to be a thing for a little over 100 years. We've only known about them for about a century. So just figuring out what was out there and what the genomes of the viruses were, how we can classify them into RNA versus DNA versus protein types and all those types of things, membrane-bound viruses, was a huge focus for a long time. Actually, a professor down the hall from me here was a guy who originally characterized herpes viruses. He was a leader in that field. And then after that, a lot of the focus started being on, okay, well, we have all these viruses that have different replication strategies. So what happens when it infects? And when they say what happens, they really just wanted to know what genes were expressed from the virus end, how many virus particles were made. If you deleted certain virus genes, how did that change the replication of the virus? So they didn't, which are all very important questions, and they've really led us to a whole new era now in the study of virology and of human diseases. Yes, it does. It applies to like any domain. This is how you have to study things. But now we're at a point in virology where we know there are hundreds of thousands to millions of viruses. There was actually a study recently, a genomic study that used DNA sequencing and discovered what I think they were estimating it was like 10 billion viruses on the world, and it's stuff that you wouldn't always see floating around an actual virus particle. They're just old virus genomes that are in different organisms. So like in human cells, the fact I gave earlier is about 20% of our genomes are actually ancient virus DNA. So we're at this era now in virology where we know that there's so many viruses, and we know that viruses are so good at replicating. And now we just want to know, or not just want to know, but what I really want to know now is like, okay, so we have these important human pathogens. We know they're good at replicating in our cells. We have no antiviral therapeutics for them or vaccines for them. So what are they actually doing to the cell? Not just what the virus is doing, but what the cell is doing. And thank you so much for that compliment. I really appreciate it. Yeah, so I'm at this interface really between cell biology and virology, where I'm not really interested in just the virus for the virus' sake, but I'm interested in how our cell is responding during the virus infection. And this takes many different venues. So, of course, if you're looking at something that's spatially organized, you want to be able to see it. So a lot of my work deals with microscopy. So I will label different compartments in the cell, such as the ER or the mitochondria, and I'll also label a virus, and I'll watch the virus enter the cell and actually watch the cell for all of infection time to see how those organelles are reorganized and how the virus is doing that. Then another thing you can do is, to get back to the gene expression problem, is use a technology called mass spectrometry, which is what the lab I work in actually specializes in. It's I haven't mentioned this yet, but I work in the lab of Ileana Cristea at Princeton. Give her a Google. She's an awesome lady. She's a world leader in proteomics and also in virus infections, and specifically virus-host interactions. But so I can apply this technology, mass spectrometry, to study how proteins are also either spatially organized throughout infection or how their levels change throughout infection. So instead of just looking at raw output of genes, of viral genes during infection or of host genes, I can start to gain insight into how proteins are changed throughout infection. So after genes have been made into proteins, if the proteins change in localization somewhere, which would mean they have a different function throughout infection time, or if maybe they have different post-translational modifications that would change their function. So by bridging both microscopy and mass spectrometry, I can start to get kind of a holistic view of subcellular organization throughout infection time. And I really, truly think that that type of work has huge implications, both for virology studies and for our just raw understanding of how the cell works. yeah, Louis, we're going to put this on YouTube, so you can go watch it later. So, does anybody have any more questions about anything? We could talk about vaccines, we could talk about herd immunity, we could continue talking about cell biology, as that's my original love. I was thinking that I would like to go over herd immunity in the last couple minutes that I have here. So we've been talking about virus replication and how viruses are so good at manipulating your cells. They can completely reprogram a cell that has normal function to all of a sudden a cell that is now entirely a virus factory, right? So this is scary. Like, we don't want this to be happening in us all the time. We come into contact with millions of viruses a day. They're good at killing us, and so we want to combat this. And so one of the ways that we've been combating this for a little over 100 years is by vaccines. What is hepatitis B vaccine's adult dose? I don't know what exactly you mean by that question. Can you rephrase it for me, please? Yes, okay, excellent question. We have much in common, I think, and Rio 7. So microtubule transport is absolutely, like, essential for virus entry and exit, things like that. Herpes viruses actually completely reorganize microtubules during their infection cycles, both to get the capsid to the nucleus and also to get mature virus particles out of the cell. But, of course, the actual, like, subcellular and temporal organization of that isn't known. It's just known that they're important, which, of course, microtubules are important, right? Microtubule-based transport of mitochondria should be important during infections, but as far as I know, nobody's looked at that. One observation our lab has made, actually, during infection in a recent publication is that mitochondria during infection become very motile. So they're normally relatively stable, and then during infection, they start to just, like, bounce all over the place. So something's going on there, and it might have something to do with the metabolic reprogramming that viruses cause. Thank you for joining us, Louis. Let's see where the other questions were. But yeah, I love looking at organobiology and things like that in the context of virology, and collaborating between fields is an absolutely essential part of doing that. Any insights from how quantum mechanics can be applied to cell biology? So I know there are some, this is not in virology, but there are some research groups that are really interested in looking at how you can model cell organization. And by cell organization, I mean like actually like the molecule level. So not at the organelle or the protein level, but at like the atomic level in cells. And are wondering if there are some underlying physical principles that would dictate that organization. So that is a very up-and-coming area of research, I would say. I don't actually study that, so I could point you in the right direction if you wanted to talk to somebody about that. Yes, also going back to the microtubule question, motors as well. So in the case of HCMV, it's known that dynein is required for assembly complex formation, but again, that's a thing where it's like, oh, if we get rid of dynein, we lose this assembly complex formation, but they don't actually know how or exactly what it's doing. It's also thought, I believe, that acetylated microtubules specifically are important during infection, and I believe that goes back to the recent studies that found that acetylated microtubules dictate the stability of microtubules, but that would also be very interesting to look at. Yeah, you should, T-Dollars, you should look into quantum mechanics in biology, because there's been a lot more bridging between fields recently, which is very important for the future of research, and there's actually a physicist that's on my thesis committee who does study that. So if you were interested in talking to him, I could point you in the right direction for sure. Yeah, I would love to talk to you more about this, Andrea. I don't know if that's how you pronounce your username. Okay, so that was all the specific content I had for you guys. Oh, like I said, I would like to go through herd immunity really quickly because I think this is a growing concern in the world, just in the world right now and in the social environments. So most likely, most of you have heard of herd immunity, and maybe you do know what it is exactly, but if you consider, so let's take, for example, measles virus. We haven't talked about this virus yet, but it is considered the most infectious human virus that we know of. So it is like an absolute master of reprogramming human cells to produce measles virus. It's kind of scary, really. It can very quickly. So herd immunity is the phenomenon where a bunch of individuals in the population get vaccines, so they're no longer able to be infected by a wild-type measles virus in this case. But you always have individuals. So first of all, to be immune to a virus, you have to have an immune system, right? You have to have an active immune system that's constantly suppressing this virus infection. But if you have some individuals in the population that maybe never got vaccinated, or they actually just can't get vaccinated, for example, immunocompromised patients cannot get a new vaccine because it would kill them, or people like very young children. You can't give, some vaccines cannot be given to children until they're above the age of like 2 or 3. Or you have someone going through cancer who is immunocompromised while they're going through chemo, and so if they were exposed to a new virus, they wouldn't be able to fight it. Or if there's a flare-up of another virus, they wouldn't be able to fight it either. So you need herd immunity to keep those people safe. So everyone in the population, except for those individuals, is vaccinated. They do not carry a wild-type virus, so other people can't get infected. But if you start to have less people that are vaccinated, then the few people in the population who cannot be vaccinated start to have a higher probability of being exposed to that. And so, therefore, they have a higher probability of getting sick. And then the further issue from that is if you have more people in the population getting sick, viruses mutate very quickly. This is obviously a huge problem with the influenza vaccine every year. We try to engineer a vaccine that will work, but influenza is such a volatile virus that even with a ton of effort of making vaccines, it's going to mutate past that and still be able to infect you. So if you have a bunch more people in the population getting sick, then there's a higher probability that mutant viruses will arise. And then those mutant viruses might be good at infecting the people who are already vaccinated to a previous strain of the virus. So as the virus evolves, right, your immune system isn't evolving nearly as quickly, and you're going to need another vaccine to combat this. So then that really undermines herd immunity, because now even the people who are vaccinated no longer have resistance against this new virus strain. So this actually is thought to be, this is the theory with a lot of support that is underlying the current increase in outbreaks of measles. In the UK, for example, I think it was a 1,600% increase in measles outbreaks in the last 2 years, which is kind of scary. There was actually in New Jersey, where I live, there was an outbreak at a children's hospital a couple weeks ago, which was also pretty scary because a lot of the children there were sick, and they were in the class of either too young to get a vaccine or immunocompromised, like I was just talking about. So they were at the complete mercy of the virus, and this killed, I think, like 20 kids. This was like a month ago in New Jersey. All that to say, it is very important that we keep herd immunity going and that we vaccinate for diseases that should be vaccinated against. That is a great question. So I'm not sure there's a specific answer to that. So, for example, flu outbreaks, right? There was the huge outbreak in 1918, which was about 100 years ago, that killed tons of people. It was a very scary strain. That one actually developed because it was a hybrid of other organisms, influenza genomes, that then got into humans, and was especially scary because it was during World War I, and you had a lot of mixing of different populations that had different immune signatures that hadn't seen each other before. There's also periodic outbreaks of the Black Death, which is a bacterial disease, not a virus infection, but similar concept. I don't think that you could necessarily put time as a definable variable that would cause other outbreaks. So by that I mean, like, of course, more evolution would have occurred over a certain amount of time, but we do not understand enough about that in the case of specific virus genome evolution to say, like, yes, if you gave it 5 years, you should have another really bad outbreak. It's a lot more random than that, and you can't really define it in that type of time. That being said, we know that the common cold and influenza come out every season, like every winter. Actually, right now in my area, it's flu season, so a lot of people are sick with it, like usual. The pattern is thought, great point, yes, the pattern is thought to be more of exposure. So Going back to the 1918 flu epidemic, that one was particularly bad both because of the new genomes we were being exposed to, but also because the adult population had never been exposed to even a similar outbreak of that, so they had no immune signature against it. Whereas the very old population, which is what's usually threatened the most by influenza, had seen a similar virus a generation before that. And so they had more of a defense already built up against influenza. So it would be correlated to some extent. And I saw a talk recently this summer and at the American Society of Virology conference where a group was doing a lot of computational analysis of outbreaks across history and trying to get more of a handle on the link between exposure and time. And of course, time is obviously important. It's just more of defining the actual time space is not quite as clear. And there's also things now that are being tossed around, like forget exactly how they're coining it, but the concept basically of exposing yourself to new virus infections from inside you and how that maybe makes you more or less susceptible to future infections. But that's a really challenging aspect of virology right now because we know that viruses are so good at infecting you, but that doesn't happen all the time. And so that's to do with things like we've talked about with the spatial organization of cells during infection, which is what I study, and also about just population dynamics and things like that. So, excellent question. All right, so if there are any more questions, I will maybe hang out for another 2 minutes or so, and then otherwise that's pretty much all I had for you guys today. So for people who came in late, my name's Katelyn. I'm a PhD student at Princeton in New Jersey. I'm about halfway done with my PhD, which is exciting. So far it's going pretty well. I've already had a couple papers published, which you can find on PubMed if you search for Katelyn Cook. They're all to do with the spatial organization of virus infection, which is what I'm super passionate about. I've been able to give a few talks at conferences. I am super happy with the PhD program here at Princeton. It's, in my opinion, the best in the country, perhaps the world, not only because the science is so good, but because we're all actually really happy here. So I have a really tight-knit community of other graduate students and professors and postdocs and people just in general in Princeton and beyond that are collaborating with me and supporting me on different things that I try. And of course, like being able to discuss science with them and get different perspectives on things is absolutely essential to any research. So yes, I love Princeton. If anyone is thinking about applying to grad school, feel free to reach out to me because I would love to give you advice on that. I never thought I would be an Ivy League student. That's like very opposite of my personality. I'm from Colorado. I went to a state school my whole life. But going to grad school here has been very rewarding. So I would highly recommend it. I really hope you guys enjoyed this discussion and you learned something. Feel free to DM me if you have any future questions. I love this stuff, so I love talking about it, and I like sharing my research with other people, both scientists and non-scientists. That was a good question. So this last question, so people have also been looking into that, but children, so when they're born, it is known that infants inherit, because they've been sharing blood with their mother, right? They inherit some of the mother's immune system, but that only stays with them because that's just the blood, not the actual memory. It stays with them for a couple months. So this is why infants have to wait to get vaccinated. So you cannot vaccinate a child right when it pops out of the womb because it won't learn anything because it still has the mother's immune system. If you do vaccinate a child too early, there's a very good chance that the vaccination won't actually stick. But the actual ability to pass them along to adults, they have that in that they are carrying a virus if they're sick with it or if they're infected with it. I don't know if there's any evidence that suggests that they are more, if they're better at passing along infections than other adults. That's something that I would have to look into more in that case. All right, everyone. Thank you for joining me. I'm going to go get a little bit of work done and then go enjoy the holidays. So I'll catch you later. Bye.